Hello, my name is Joshua Mugabi. I welcome you to Biology Remedial Lessons. Today we are on Remedial Lesson Part 5. And this lesson we are going to talk about cells. You are welcome. Define the term cell. Define the term cell. We can say that it is the basic unit of organization of an organism. It is the basic unit of organization of an organism. And that is the basic functional and structure unit of an organism. So the cell is the basic unit of organization of an organism, which is the basic functional and structure unit of an organism. What is cell biology? What is cell biology? Cell biology is the study of structure and functioning of a cell. Is the study of structure and functioning of a cell. It is also called cytology. It is also called cytology. Draw the genostructure of a plant and animal cell. Draw a genostructure of a plant and animal cell. When you look, those are our structures. On your left, we have the plant cell, and on the right, the animal cell, with different parts: chloroplast lining the cytoplasm, endoplasmic reticulum, nucleus, thick cellulose wall, cell membrane, ribosomes, mitochondria, central vacuole, insert, Golgi bodies. And most of those ones you can also find them in the plant cell. List the parts of a cell that can be seen under an electron microscope and state the functions of each part. Want to list the parts of a cell that can be seen under an electron microscope and state the functions of each part. We shall start with the cell wall. The cell wall is found in plant cells in addition to cell membrane. It is found in plant cells in addition to cell membrane. The cell wall is made of cellulose, which makes the plant tough. It's made of cellulose, which makes the plant tough. It also allows gases and water and other substances to pass through. It allows gases, water, and other substances to pass through. Cell membrane. The cell membrane is permeable or selective permeable to control movement of materials in and out of the cell. The cell membrane is pressed against the cell wall, and it is permeable or selective to control movement of materials in and out of the cells. It bounds or encloses the cell contents. It bounds or encloses the cell contents. It is also called plasma membrane, or sometimes called plasmalemma. It is called a plasma membrane or sometimes plasmalemma. The cytoplasm. The cytoplasm is a fluid medium where chemical reactions occur. It is a fluid medium where chemical reactions occur. Also, where cell organelles are suspended. In the cytoplasm, it's where we find cell organelles suspended. We have the nucleus. The nucleus is composed of the nucleolus, nuclear envelope, and chromatins. So we can say that the nucleus controls cell activities. The nucleus is the part of the cell which controls the cell activities. The nucleolus is found inside the nucleus, and it is where synthesis of DNA takes place. In the nucleolus, it's where the synthesis of DNA takes place. We have the vacuole. A vacuole is large with a central position in plants. This is a sac filled with fluid called cell sap. A vacuole is a sac filled with a fluid called cell sap. It is large in plants but small in animals. It's large in plants but small in animals. It also acts as a reservoir for food and harmful waste which would otherwise interfere with the metabolism in the cytoplasm. The vacuole acts as a reservoir for food and harmful wastes which would otherwise interfere with the metabolism in cytoplasm. We have the lysosomes. Lysosome, these ones store hydrolytic enzymes. They store hydrolytic enzymes. They destroy worn-out cells and organelles, cells, pathogen, and many others. The lysosomes destroy worn-out cell organelles, cells, and pathogens. They also take part in digestion of food in unicellular organisms. They take part in the digestion of food in unicellular organisms. Those are the lysosomes. They are autolysis. Lysosomes are autolysis. We have the Golgi apparatus. The Golgi apparatus, these ones process and package of synthesized material. They take part in the processing and packaging of synthesized materials. Those are the Golgi apparatus. They also are involved in the transporting stroke secretion of packaged cell materials. They are involved in the transporting and secretion of packaged cell materials. Example, the glycoproteins and the mucus. They are also involved in the production of lysosomes. They are also involved in the production of lysosomes. Those are the Golgi bodies. We have the ribosomes. Ribosomes, this is where protein synthesis takes place. In the ribosomes, it's where protein synthesis takes place. The mitochondria. In the mitochondria, it's where the synthesis of ATP or energy takes place. The mitochondria synthesizes ATP or energy. We have the chloroplasts. Chloroplasts are found in plants. And in the chloroplast, it's where photosynthesis takes place. In chloroplasts, it's where photosynthesis takes place. We have the endoplasmic reticulum. The endoplasmic reticulum is involved in the transport of cell secretion. It is involved in the transport of cell secretion. That is the endoplasmic reticulum, involved in the transport of cell secretions. It can be rough or smooth. So we have the rough endoplasmic reticulum and also the smooth endoplasmic reticulum. State the functions of the cell sap. State the functions of the cell sap. The cell sap stores chemical substances such as sugar and salts. And the cell sap, we find it in the cell vacuole. It stores chemical substances such as sugars and salts. It maintains the shape of the cell and also provides mechanical strength. The cell sap maintains the shape of the cell and provides mechanical strength. It also plays a role in osmoregulation by creating an osmotic gradient that brings about movement of water. The cell sap plays a role in osmoregulation by creating an osmotic gradient that brings about movement of water. Compare plant and animal cells. Compare a plant and an animal cell. Plant cells have chloroplasts, which is lacking in animal cells. Plant cells have chloroplasts, which are lacking in animal cells. Animal cells have many small vacuoles, while plant cells have a small central vacuole. Animal cells have very many small vacuoles, while plant cells have a large central vacuole. Plant cells have cellulose cell walls, lacking in animal cells. The plant cells have cellulose cell walls, which is lacking in animal cells. Cytoplasm in plant cell is in the periphery, but in animal cell it is centrally placed. The cytoplasm in the plant cell is in the periphery, but in animal cell it is centrally placed. Plants store starch, oil, and proteins, while animals store guts and glycogen. Plants store starch, oil, and protein, while animals store guts and glycogen. Animal cells have centrioles, which plant cells do not have. Animal cells have centrioles, which plant cells do not have. Explain the meaning of each of the following. Explain the meaning of each of the following. Cell. A cell is a basic unit of organization in an organism. A cell is basic unit of organization in an organism. Specialized animal cells include the sperm, ovum, and muscles. Some of the specialized animal cells include the sperm, ovum, and muscles. Specialized plant cells include the epidermal cells of the upper and lower epidermis, the guard cells, mainly on the lower epidermis, and the palisade cells on the upper side of the leaf. Tissue. These are cells of a particular type grouped together to perform a certain function. Tissue are cells of a particular type grouped together to perform a certain function. Animal tissues include epithelium, blood, nerves, muscle, skeletal, and connective tissues. Animal tissues include the epithelium, blood, nerves, muscle, skeleton, and connective tissues. Plant tissues include the epidermal tissues, photosynthetic tissues, vascular tissue, and strengthening tissues. The plant tissues, they include the epidermal tissues, photosynthetic tissues, vascular tissues, and strengthening tissues. The organ system. Organs are grouped together to form systems, also called organ systems. Organs, when grouped together, they form systems which we call organ systems. Plant systems include the transport system. The plant system includes the plant system which we call the transport system. Here we have the xylem and the phloem. Xylem vessels, phloem sieve tubes. Animal systems include the excretory system, digestive system, respiratory system, nervous circulatory system, endocrine or hormones and gland system, and skeletal system. Animal system includes the excretory system, digestive system, respiratory system, nervous system, circulatory system, endocrine or hormones and gland system, and skeletal system. Name the structures which are present in plant cells but absent in animal cells. Name the structures which are present in plant cells but absent in animal cells. We have chloroplasts and cell wall. Chloroplast and cell wall. Name the structures which are present in animal cells but absent in plant cells. Name the structures which are present in animal cells but absent in plant cells. We have the lysosomes, centrioles, pinocytic vessels. We have the lysosomes, centrioles, and pinocytic vesicles. Explain how to estimate cell size. Explain how to estimate cell size. Cell sizes are measured in units known as micrometers. The cell sizes are measured in units known as micrometers. Required is a transparent ruler marked in millimeters. To estimate the cell size, we need a transparent ruler marked in millimeters. One micrometer is equivalent to 1 millimeter divided by 1,000. To get 1 micrometer, we get 1 millimeter and divide it with 1,000. Then we have 1 micrometer. The procedure. You click to low power. Click to low power. Then place the transparent ruler with its millimeter marks on the stage. You place a transparent ruler with its millimeter marks on a stage as shown in the diagram. Place the transparent ruler with its millimeter mark on the stage. Focus so that the millimeter marks can be seen as thick dark lines. You focus so that the millimeter marks can be seen as thick dark lines. Now estimate the diameter of field of view by counting the 1 millimeter spaces between the first mark and the last one across the field of view as shown. You estimate the diameter of the field of view by counting the 1 millimeter spaces between the first mark and the last one across the field of view as shown in the diagram. The diameter of the field of view here is estimated as 3.2 millimeters. The diameter of the field of view here is estimated as 3.2 millimeters. Convert the diameter of the field of view from millimeters to micrometers. That is 3.2 divided by 1,000. We convert the diameter of the field of view from millimeters to micrometers. That is 3.2 divided by 1,000. Estimate the fraction of the field of view occupied by the cell. We now estimate the field of view occupied by the cell. This is done by estimating the number of cells, places, end to end, that would fill the diameter of the field of view as shown. We estimate the fraction of the field of view occupied by the cell. And this is done by estimating the number of cells, places, end to end, that would fill the diameter of the field of view as shown. In the figure, it is estimated that approximately 60 cells will occupy the diameter of the field of view. In our diagram, as we are seeing, it is seen that approximately 6 cells are occupying the diameter of the field of view. Therefore, one cell will occupy 1 out of 6 of the field of view. We can see from this diagram that one cell will occupy 1 out of 6 of the field of view. Its diameter is calculated as 1 out of 6 times the diameter of the field of view. The diameter of one cell is calculated as 1 out of 6 times the diameter of the field of view. In a drawing of a giraffe, the height of the head from the ground was recorded as 10 centimeters. The drawing also showed a magnification of 0.02. Calculate the actual height of the giraffe. In a drawing of a giraffe, the height of the head from the ground was recorded as 10 centimeters. The drawing also showed a magnification of 0.02. Calculate the actual height of the giraffe. We know that the drawing height divided by the magnification, which is 10 centimeters divided by 0.02, it gives us 500 centimeters. In a class experiment to estimate the size of cells, a student observed and obtained millimeter marks on the field of view of a microscope as shown in the diagram below. In a class experiment to estimate sizes of cells, a student observed and obtained millimeter marks on the field of view of a microscope as shown in the diagram below. If the student counted 40 cells on the diameter of the field of view, what was the approximate size of each cell in micrometer? If the student counted 40 cells on the diameter of the field of view, what was the approximate size of each cell in micrometer? The diameter of the field of view divided by the number of cells. This one will give us 3 times 1000 divided by 40, giving us 75 micrometer. Diameter of the field of view divided by the number of cells. And the diameter of the field of view in micrometer is 3 times 1000 divided by 40, which is the number of cells. This one gives us 75 micrometer. That is the size of each cell in micrometers. Under which of the following light microscope magnifications would one see a larger part of the specimen? Times 40 or times 400? Give reason. Under which of the following light microscope magnifications would one see a larger part of the specimen? Is it X40 or X400? Give reasons. It is X40. This is because smaller magnification gives a wider field of view, hence a larger part is seen. Where we see a larger part of the specimen is times 40. The reason is smaller magnification gives a wider field of view, hence large part is seen. Hello my friends. Thank you very much for participating in cells. Let's meet again in remedial lesson part 6, where we shall talk about cell physiology. Thank you. Please don't forget to press that subscription button. Bye-bye.